Thank you. So many of you probably do not know me. I'm Gary Luker. I'm from the medical school. But one of the things I really have enjoyed about being here at the University of Michigan is the opportunities to build bridges and collaborate from the medical school to the College of Engineering. And we've had a number of, I think, very exciting and productive collaborations across the years that we've been here that have allowed us, and hopefully our collaborators on the engineering side, to ask questions that would otherwise be impossible, at least with my expertise. And the project that I'm going to talk about, I think is perfect timing with the celebration we've just heard for Krishna, involves Krishna and also Sid, who's back there, for work that is ongoing and I think has brought a really new perspective to an important problem in cancer biology. So I give you that preamble for two reasons. One, to acknowledge the important work that Krishna and Sid and others are doing on this, and two, so that when you start delving into the computational aspects of questions, I'm going to smile and point you to it now, okay? So the premise of this is really that we want to use artificial intelligence to understand the heterogeneity in cancer. So, that's a big goal, but I'll explain why it's important. Because heterogeneity, in my opinion, is one of the major problems, if not the biggest problem, preventing us from understanding and curing cancer. So heterogeneity exists at a huge number of levels in cancer biology. One patient with breast cancer has a different tumor from another patient with breast cancer, even if we classify them both as, say, estrogen receptor positive. They're both different. Within a tumor, the cancer cells are also very heterogeneous. One cancer cell does not equal another cancer cell, and that differs both from a primary tumor to other sites of metastases. So this heterogeneity causes an immense problem in understanding cancer biology and also how we approach treatment. So as we go into the talk, I just want you to think about the problems we're really trying to understand are single cell analysis, single cell understanding of cancer to tackle the problem of heterogeneity. And I'll show you a clear example of that very early on in here. But then, leveraging expertise, Krishna, Sid, others that I'll mention throughout this, we've developed an integrated experimental and computational workflow. And it really is bidirectional, as Krishna said, also a team approach to make this work so that we can start to look at how cancer cells signal and how they migrate and understand those processes. I'll show you just an initial applications work, how we've applied it to identify a testable hypothesis that fortunately turned out to be right, and some future directions for what we're going to be able to do. All right, so if we just step back at the beginning and say, ignore everything I just said about heterogeneity, and if we just think about population scale readouts, and this is just one example of a readout we do in our lab looking at how cancer cells signal. It could be pretty much any biological readout. You can take a population scale event, that's the black line in the middle there. You can apply some stimulus, measure the response, and you look at this and say, you just spent quite a bit of time telling me heterogeneity is a problem. This doesn't look so bad. I mean, yeah, there's some error here. Maybe we don't do great experiments, and probably that's part of the error that contributes here. But overall, this doesn't look so bad.
What is the y-axis here?
The y-axis is kinase activity for a kinase known as AKT, which is important in cancer cell migration, survival. It's one of the major oncogenic kinases in cancer. And we're measuring this with a fluorescent recorder for kinase activity of AKT, okay? So the premise here, we're assuming that all these cells act the same and that they are the same. But then let's take a look a little bit deeper. So I'm going to show you the same data, but now we're looking at them as single cells, okay? Now we have a much bigger problem. You can see cells along here. Some of them don't respond at all. Others respond incredibly well. Others seem not actually drop their signal. So now we have a much bigger problem. And to understand this, this is where we want to leverage computational expertise to develop really an entirely new approach to the problem, to develop a physics-informed artificial intelligence platform to be able to identify the reward system, if you will, thinking of our cells as kind of intelligent actors, what motivates them to behave, signaling or migration, example I'll show you later, what motivates them to act as individuals and sometimes to act as team players working together and sometimes as competitors. And having the physics constraint is really important to this work because without it, the artificial intelligence approach might come up with answers that just aren't physiologic for a cancer cell. And anyone in cancer biology would say, that doesn't happen. What are you thinking? So merging those two together is very important. All right, so here's just kind of the overall workflow for how we are approaching this problem. And there's innovation on all these sides. I mean, I know more about the innovation, because we're developing it along with my wife, Kathy Luker. She and I do the experimental side of this work together, but developing the experimental approaches to image and quantify thousands of single cells over space and time and get multiplex readouts. All of that information then feeds into the physics and chemistry mechanistic models that Krishnan and Sid do to be able to infer what are the physical material properties of these cells, how are they acting? And then to use that information to understand some of the transition probabilities for inverse reinforcement learning. That's our artificial intelligence platform to be able to understand how these cells work. And the goal is then to make predictions. Predictions based on observing how the cells behave and understanding the physics of how cells do so. We then want to test those interventions, and you can see various feedback loops, that we're providing information to refine the models, to come back and say, well, we need more data. We need these specific experimental data. All of these to be able to apply broadly to biomedical research. So our initial challenge was looking at cancer cell migration. So this is a fundamental problem in cancer. Migration and invasion really define important steps in cancer biology. For an epithelial-based cancer like breast cancer, invasion beyond the basement membrane, the initial constraint for those cells, is a key distinction in situ versus invasive cancer. And already in that literally millimeters of movement, prognosis has already changed dramatically. And then invasion is also important in metastasis, spread of the cancer to different sites. And metastasis, by and large, in not only breast cancer, but almost all malignancies, is what causes death. And really, our fundamental challenge in overcoming invasion and metastasis is really, there still isn't an overarching understanding of how heterogeneity occurs in migrating cancer cells. So I'll just show you a few high points. I'm not going to go into tremendous details about how we get this information, but as we're going to do these measurements, we're going to collect information from hopefully thousands, working to get more, individual cancer cells as agents. And we're going to measure, we're measuring several things. In panel A, we're measuring the kinase that I talked about, AKT, as well as others, and we're going to measure the dynamics of signaling by those pathways over time in single cells. We're going to look at changes in cell morphology, because those are very critical in understanding processes associated with invasion and migration. We have automated image analysis software to be able to track all this information. And then to get higher order information, then we start to look not only at the magnitude of the signaling, but the frequencies, the oscillations of this, because oscillations provide additional layers of information as to how cancer cells and all cells in general really interpret signals and then act upon. And from that information, then we're going to provide those inputs plus others to the computational modeling approach. And really because we'll see this a little bit later in terms of our testable hypothesis, we just use a very simple microfluidic device, use a chemoattractant, either CXCL12 or EGF. Those are just important molecules driving migration and invasion of cancer cells, among other phenomena. And then using time-lapse live cell microscopy, we can image those cells as they move, track their single cells over time, and then we get a display kind of truncated only to 250 cells here. But you can see heterogeneity both in their signaling, the distance they move, red in this plot is toward the gradient, blue is cells that kind of get lost and go the wrong way. We're still working to understand that, but how far they've moved, and more importantly, how closely do they reflect movement toward the gradient. So these are the information that we have. This is a lot of information. We need to know what to do with it. And that's where we now turn to the approaches that can take this information, put it into a physics background, and then link it to inverse reinforcement learning. And this is work from Christian and Sid, so I feel woefully inadequate to try to explain it all what they do. But suffice it to say that this is a really, I think, fascinating computational framework to be able to infer the physical mechanisms that cancer cells are using to migrate or do other behaviors expressed as partial differential equations. And you can read faster than I can talk. So basically, I'll just summarize in that this approach considers the cells as they're behaving as materials, and looking at the densities of these materials over time, they can go through and then infer what are the physical properties that are governing these behaviors, and select really the one that fits best. And this can capture the physics of cell migration. And I'll just show you one illustration of this, a fairly simple one, but I think important in terms of how, at least we on the experimental side really thought about this, just the ability of cancer cells to migrate on a relatively soft versus a relatively stiff substrate. It's important in cancer because tumors typically are stiffer than normal tissues, and those mechanical properties play an important part of cancer biology. I mean, we made the observation experimentally that the cells migrate more on a stiff substrate. But then using the VSI approach with Christian and Sid, they considered a variety of mathematical approaches that could explain what we were seeing. And interestingly to us, and I think a discovery that is new to the field, is that really the difference in the ability of these cells to move more is based largely on their ability to proliferate in the stiffer environment, and that their actual movement difference is much less. So that's really a physics-informed approach, taking the experimental data we give and say, here's an underlying mechanism that explains it. So then the second component of this approach is inverse reinforcement learning that Xuan Han and Nicola Banovic are really leading, well, students in their group, Chen Yang, for example. And this is an approach, it's a data-driven method to infer and understand the rewards that an agent is seeking. So you watch the behaviors and then try to understand what are the motivations for that behavior. So for example, in a room like this, someone using this approach might come in and say, why does everyone sit in the back? Is the speaker kind of toxic? Is he scary? I don't know. So that could come up with some really strange explanations that we have to constrain. But you get the point in that this is a way to observe a situation and try to understand the rules, the policies that govern that behavior. And the key terms to think of in this is the state, what's the current condition? All right, the action, what's the next step that this agent is going to take to seek its reward? The policy, what is the strategy underneath it? What informs the agent and the next steps? And then the reward is obviously, okay, what's the big outcome? It can be a positive reward, moving towards something like CXL12, a chemotherapy in our case, or it could be getting away from a hazardous situation or a bad situation like being in a crowd for cancer cells who want to try to move. All right, but one of the major limitations for our concepts of cell migration is that IRL, you need to know the transition function that governs all those transitions in state policy action beforehand. But Christian and Sid, working with Xuan, came up with a way that VSI can then infer and understand that transition probability through the Fokker-Planck equation. All right, and that's where if you ask questions, they're the ones to answer. But I think this is really cool because now we have informed that key step in the agent, the cell, decision-making process. It helps us understand the motivations and reward structures. and sets that in the context of physics of cancer. All right, so this is just kind of an illustration that helps me, if no one else, to understand, what this framework does. So, I mean, you've got heterogeneous cancer cells moving toward their reward. Cancer cells may not be interested in money so much as the chemotactic gradient, but on all those steps, then, the markup decision process in IRL considers, the various states, actions, based on some changes that we can have, signaling, morphology, nuclear position, and then considers what's the cost for the next step in the process, what's the cost for the action. And that's where the VSI FP framework can provide those probabilities and help us understand the actions that may occur. And this is just one example of how we've used this. So if we're thinking about cancer cells migrating toward a chemoattractant gradient, in this case, we're only looking at the amount of AKT activity in panel A, so that kinase I showed you early on to start with. And then if we use the FP-IRL framework, we can infer that the most likely scenario, the state that the cells want to be in to take the action of moving toward the chemoattractant gradient, is cells that have a medium-low level of AKT activity. So this is just the data we've given them, observing the outcomes and making that prediction. And this is just showing the probabilities of velocity toward the gradient for the different conditions. So this predicted something that we could test, that if we can drop the AKT activity of a cell, that it should migrate more. So this is something we can do. So experimentally then we said, if we treat with alpelisib, alpelisib is a drug that's approved for breast cancer and other malignancies. It inhibits upstream of AKT and blocks that activity. So then if we do the same experiment that we showed before, and then if we quantify the migration velocities toward the gradient, if we inhibit with alpelisib, we end up with more cells that move toward the gradient more effectively, so better directional migration. They have fewer cells that actually go in the wrong direction, which I think is quite interesting in and of itself, and they move faster. So, obviously there are lots of details in the process, both the experiments and the computation, that are completely glossed over here in the interest of time. But this is the approach that we're using to be able to understand from our observations mechanisms and testable hypotheses. Then we can come back to and say, here's a way we can change the behavior of these cancer cells. All right, so in summary, we really developed for the first time a VSI-informed framework for cancer cell migration, which I think is really quite remarkable. From the computational side, I mentioned how VSI has now provided the transition function, which is essential for IRL. And this overall framework constrains the artificial intelligence to the physics that we can observe and measure of cancer cell behavior. And, this is early on in the work, but we've already identified a testable hypothesis and validated it. And now we can go back and build on this. And how are we going to do that? So right now, we just had one cell type, cancer cells, moving in one direction. Tumors are not just cancer cells. In many tumors, cancer cells are actually a minority population. There'll be fibroblasts, immune cells, a variety of other cell types. So now we really want to progress in the future to understand multi-agent interactions. How do these cells work together? How do they cooperate? How do they influence each other's behavior? Which obviously creates more computational problems in terms of the scale of the work. And also on our side, we have to get a lot more experimental data. So we're working toward that. And based on this framework, we're happy to, based on funding from MICD and MIDAS, we're able to put together a new NIHR1 grant based on this. Krishna has assured me that he's going to continue working on this project for at least the next 24 years, which may outstrip my time doing the project, but I'm happy that we'll be able to continue with Krishna and Sid on this work. So this is the project team. I've mentioned most of the people along the way. Jennifer Lindemann also contributed to the proposal. My wife, Kathy, Kenneth, Chen Yang, Patrick, and Sid. all experts. This is my cat, who I feel I have put in every presentation I give. Really want to thank the internal funding sources, and also just kind of a shout out and thank you to MICD and Midas for funding us. We did get external funding from the William Keck Foundation for this work. I will say that even though other teams from the University of Michigan had won the Science Award, we were the first University of Michigan team to win the Medical Science Research Award from the Keck Foundation. So I think that really attests to the novelty of the work as they perceived, and I think the impact.
Thank you very much, Greg. That was excellent talk, and also really nice to see how multidisciplinary it is, and very happy to be there to get these things off the ground.
Thank you.
Yeah, I have a couple of questions, but I want to see if audience wants to start off with some questions.
The first thanks for the very interesting. And also I want to learn more about the environment. So like you mentioned, after you get some action, you will need to have some reward function or support, right? So in order to get this environment, are you using the simulation environment that you already know about the physics knowledge or something, or are you doing the experiments to collect the data next to the different trials?
So at this point, what we're doing is using systems that we think we understand on population scale. I mean, we've worked on the CXL12 pathway for a long time. We know the cancer cells migrate toward it. What's, on a bulk scale, what the new information that we're learning is providing more detail on the single cells as they move. How are they signaling? How are they changing their morphologies? So right now, the innovation on the experimental and cancer biology side is really the single cell analysis, providing the data for the computational side. As we move forward, as we start to think about multi-agent scenarios, different cell types, then we're getting into some areas of cancer biology that are not as well understood. So, the biology will be less well known, and I think we'll come up with some things that are quite interesting. Thank you.
Yeah, so I was curious, the experiments, what is the diagnostic technique and what are the uncertainties there? How do you think that affects some of the decisions?
So all these are done by live cell fluorescence microscopy. So we have multi-channel fluorescent readouts that we use for them. Because we're tracking so many cells over space and time, this is automated image processing that Kathy's developed. We do manual curation of it. Is it perfect? No. Is it pretty good? Yes. So our errors are relatively small in terms of the measurements we make of the kinase activities and the cell shapes.
Okay, very cool. Yeah, thank you very much again. This is fascinating work, and very good luck for all these further plans you have for the